Let's take a look at our world population. Today, it is estimated that there are 7.9 billion people in the world. That's over 300% more than the 2.5 billion that were around in 1951, fewer than 100 years ago. Global population scientists estimate that by 2050, that number will have increased to 9.7 billion. And that means a whole lot more mouths to feed. Currently, wheat makes up 20% of the world's food source energy, and 25% of the dietary protein that we consume. If population growth increases as expected, wheat demands are projected to rise a whopping 1.7 tons per hectare. In terms of food, that's enough wheat to produce 1,000 loaves of bread for every acre of wheat-producing land in the country. That adds up to about 50 million loaves produced in the United States alone. Another primary food source is rice. Rice is the primary food source for half the world's population, and the global rice demand is expected to rise by 59 million tons by the year 2035. Climate change and world population growth demand solutions to increase crop yields and the sustainability of agriculture. Contemporary breeding techniques and the rudimentary mutagenesis used today are unpredictable, costly, and time-consuming, and risk not being able to keep up with the growing population. CRISPR-Cas9 is an advanced genome editing technique that could be utilized to solve the projected food demand problem. So let's take a dive into just how CRISPR actually edits these genes. It all starts with good old deoxyribonucleic acid, also known as DNA, and in our case, Twizzlers and Sour Patch Kids. You might have heard about thymine, guanine, cytosine, and adenine in your Bio 101 class. Sometimes they get in a bad order with each other, and that can lead to some bad problems. It's like when those two kids that don't get along in your class stand next to each other in line on the way out to recess. It never leads to good results. The genetic sequences can make a plant weak or unsuited for its environment. This is where CRISPR comes into play. CRISPR is an acronym for clustered, regularly interspaced, short palindromic repeats of DNA. These groups are repeated DNA that are led by a specifically designed guide RNA. That guide RNA leads a Cas9 CRISPR-associated protein to the sequence. This is like the tour guides that make sure you don't get lost when you're trying to find the bathroom. The RNA lines up like a perfect puzzle piece. Then it splits and slices the DNA apart. The bases are then modified, deleted, or added to. Simply cutting the DNA can inactivate gene expression. Then, new segments of DNA can be inserted to give new traits and abilities to the plants, like the ability to fly. Or maybe something a little more realistic. One large advantage to CRISPR is that it's very time efficient when compared to the other main method of gene editing. Plant breeding can take 6 to 15 years to create a better variety that is worth producing. CRISPR also has a low cost when compared to contemporary gene editing techniques such as TALENs or ZFN. The precision of CRISPR is also one of its main strengths. Precise editing helps to avoid linkage drag, which is when a bad gene is inherited with a good one because they are near each other in the strand of DNA. CRISPR is not without its downsides, however. Critics of the technology claim that by allowing novel organisms with uncontrolled traits into the environment, the biodiversity of the entire ecosystem could be threatened. Additionally, by creating cropping systems that resist certain diseases and their vectors, the opportunity for outside diseases to gain control is a distinct possibility. Due to the potential side effects, governments around the world are beginning to put in place regulations on the application of CRISPR in crop breeding programs. Efficient and effective agriculture influences public health and can make the difference between people being able to afford another week of groceries or going hungry. It's worthwhile to consider if CRISPR is a wise tool to use to advance agriculture. We have seen how DNA can be spliced and subsequently modified or deleted to change plants. This has many positive outcomes, like higher crop yields, but it also tampers with nature. So now that you know a little more about CRISPR, you can decide if it's a healthy step forward.